Chromosomes are the heredity vehicles of the cell. They contain the heredity information that is passed on from parents to offspring. Biochemically, they are nucleoprotein bodies that stain with basic dyes. Chemical analysis shows that they are primarily made of DNA and proteins with a small amount of RNA. The proteins are of two major classes, basic proteins called histones and non-histone chromosomal proteins. In a dividing cell, chromosomes can be observed clearly under a microscope. They carry genes arranged in a linear order. In prokaryotes, genetic information is stored in the nucleoid, which in turn contains a single molecule of DNA or RNA. Whereas in eukaryotes, the genetic information is stored in several chromosomes. When a cell is in the interphase stage, its genetic material is in the form of a loose network of nucleoprotein fibers called chromatin. Under an electron microscope, the chromatin fibers appear like beads on a string. The beads are nucleosomes. A nucleosome is made of two copies, each of 4 histone proteins with DNA wound around them. During the prophase stage of cell division, the chromatin threads condense and transform into compact structures called chromosomes. Different regions of the chromosome stain differently. Lightly stained regions are called euchromatin. These contain loosely packed chromatin fibers. Most active eukaryotic genes are located in the euchromatin region. Tightly coiled, darkly stained regions are called heterochromatin. Heterochromatin regions are genetically inactive and contain non-coding DNA. The DNA in a chromosome is highly supercoiled. You will be amazed to know that if the chromosomes in any one cell of your body are unwound, they will form a long thread of DNA, approximately 2 meters long. The number of chromosomes for each species is constant. Humans contain 23 pairs, that is, 46 chromosomes. Elephants have 56 chromosomes, while a mango tree has 40 chromosomes. The number of chromosome sets in a cell is called its ploidy. A single set of chromosomes is denoted by N, and the cell is called haploid. If there are 2 sets of chromosomes, the cell is called diploid, 2N, and so on. A human body cell is diploid because it has 2 sets of chromosomes. Each set comprises 23 chromosomes. However, germ cells are haploid, as they have only 1 set of chromosomes. A chromosome is a thread-like structure composed of chromatin. It is divided along its length into 2 identical strands called sister chromatids.
The centromere is the primary constriction of the chromosome, where the sister chromatids are held together until anaphase. A disc-shaped protein structure called the kinetochore is attached to the centromere. The ends of the chromosome are called telomeres. Some chromosomes show secondary constrictions. When present at the distal end of their arms, these terminal bodies appear sac-like or like satellites. The chromosomes bearing them are called sat chromosomes. They are inactive regions of the chromosome and contain non-coding DNA. Based on the position of the centromere, chromosomes are classified as metacentric, submetacentric, acrocentric, and telocentric. In the metacentric chromosome, the centromere lies midway along the length of the chromosome. The two arms are almost equal in length. It appears V-shaped during anaphase. In a submetacentric chromosome, the centromere is a little away from the middle region of the chromosome. Therefore, one arm of the chromosome is slightly longer than the other arm. It appears L-shaped during anaphase. In an acrocentric chromosome, the centromere is located very close to one end of the chromosome. Therefore, one arm is very long and the other arm is very short. It appears J-shaped during anaphase. In a telocentric chromosome, the centromere is located at one end of the chromosome. It appears rod-shaped during anaphase. Each species of organism can be identified by its characteristic set of chromosomes. When chromosomes are arranged according to their length and position of centromere, the group of characteristics that identifies a particular set of chromosomes is called the karyotype. Karyotype analysis is important to detect any genetic abnormalities in an unborn fetus. Karyotype analysis of the fetus can be done by amniocentesis. Chromosomes contain genetic information in the form of DNA. As the vehicles of heredity, they determine the characteristics of the cell and its progeny. DNA contained in the chromosomes is responsible for the synthesis of proteins. Chromosomes play an important role during cell division. Recombination of genes in chromosomes is due to the process of crossing over. This leads to genetic variation. Changes in chromosomes will result in mutations. Mutations and recombination produce genetic variations that make evolution possible.